Hello everyone. In this video, we are going to talk about insertional inactivation. Topic of class 12th biology chapter biotechnology principle and processes. Before beginning, let me explain you in simple words what do we actually mean by insertional inactivation. Suppose this is gene X and it is responsible for forming product X. So, what do you think if we insert any foreign DNA into this structure, will it be able to form the product X? Let's see. On insertion of any foreign DNA into this structure, it will not be able to form the product X. It will lose its expression. So, in simple words, the inactivation of gene X due to the insertion of foreign DNA into it is known as insertional inactivation. Insertional inactivation can also be seen in the terms of antibiotic resistant gene. See, suppose this is a bacterial cell and it is a plasmid. It has tetracycline resistant gene. That means it can show resistance towards tetracycline. And if this plasmid is inserted into this bacterial cell, the bacterial cell as well develop the property of showing resistance towards tetracycline. That means this bacterial cell will be able to survive in tetracycline medium. And see, this is plasmid 2. It also has tetracycline resistant gene, but in this plasmid we have inserted a foreign DNA. So what will it undergo? It will undergo insertional inactivation. That means it will not be able to show its property of tetracycline resistance. It means if this plasmid has been kept in a tetracycline medium, it will not be able to survive. And similarly, if this plasmid has been inserted into this bacterial cell, the bacterial cell will also become inactive. The bacterial cell will also develop. Bacterial cell will also lose the property, that is, it will not be able to survive in the tetracycline medium because the plasmid has a foreign DNA inserted into it and has undergone insertional inactivation. So this is all about insertional inactivation in simple words. So let us read what is the definition given in the book. Definition of insertional inactivation. The phenomena of inactivation of a functional gene due to insertion of a foreign gene within the coding sequence is called insertional inactivation. Suppose this is a vector and it has ampicillin resistant gene as well as tetracycline resistant gene, but it does not have any foreign DNA inserted into it, either in tetracycline resistant gene sequence or in ampicillin resistant gene sequence. So this vector is known as non-recombinant vector. Similarly, here is another vector and it also has ampicillin resistant gene and tetracycline resistant gene. But what do you see here? It has got an inserted foreign DNA into the sequence of tetracycline resistant gene. So this vector is known as recombinant vector. Simple words, this if we keep this vector in tetracycline medium, it will not survive because it has also undergone insertional inactivation. But if we keep this vector, it will survive because it has not undergone insertional inactivation. See, if we need to transfer this all into a bacterial cell, this is a bacterial cell, there will be three possibilities. And what are those three possibilities? It will become non-transformant, transformant that has two subdivisions, non-recombinant DNA it has received or recombinant DNA. It must be clear to you what do we mean by non-recombinant vector and recombinant vector. So see again, if this bacterial cell has not received any vector into it, so this bacterial cell will be known as transformant. And if it has received either of the two, then it will be known as transformant. But up till now, it might be clear to you that we need transformant that has recombinant vector. For this purpose, we will keep the host cell in an ampicillin medium, and the non-transformant cell will not survive in the medium that has ampicillin. But if this bacterial cell has got this vector has been inserted into this bacterial cell, so this bacterial cell will be able to survive in a medium that has got ampicillin. And the reason is it has got ampicillin resistant gene. That means it will be able to show resistance towards ampicillin. And similarly, in this case, it has also got ampicillin resistant gene, and it has not got any foreign DNA in the sequence of ampicillin resistance gene. So it will also survive in the medium that has got ampicillin in it. So what is the purpose? What is the purpose to keep the host cell in a medium containing ampicillin? The purpose is to select the transformant. And the second step, as you all know that we need recombinant vector into the bacterial cell, that is host cell. So the second step is to remove this non-recombinant vector, because it will be able to survive in ampicillin medium. But let's see how we will do it. We'll transfer it, the host cell, into a medium that contains tetracycline. So if we transfer it and the bacterial cell has got non-recombinant vector into it, so it will survive in the conditions that has tetracycline medium. But if the bacterial cell has recombinant vector and we keep the bacterial cell into a medium that contains tetracycline, this bacterial cell will not be able to survive in that condition. This is because the recombinant vector has got a foreign DNA in the sequence of tetracycline, and hence it will become sensitive and will not be able to survive. And this is how we carry out insertional inactivation, and it is used, and this is how we make the use of selectable markers. But let me tell you, this method is somewhat complicated, or it needs hard work. This is because it needs to be plated again and again. So for the ease, a second method has also been developed. It is known as blue-white selection method or coloring method. We'll discuss it later. Thank you.